So, water is extremely important in biochemical reaction. So they're able to ionize into H plus and OH minus, and it can also ionize other chemicals like acids and salts. So in aqueous solution, an acid is a substance that releases H plus ions. So H plus ions can also be called protons because they only have a single positive charge and no electrons. On the other hand, bases are substances that are proton acceptors, so they can combine with H plus ions. Another definition of bases is also that bases can release OH minus ions or hydroxyl ions. Strong acids are substances that can release all of their H plus ions in a watery environment. For example, hydrochloric acid or HCl in the stomach is a really strong acid because HCl releases all of its H plus ions in the stomach's environment. So this strong acid aids in digestion, and it also helps in killing the microbes that we ingest along with food. And bases, on the other hand, are proton acceptors. Now, our body has a perfect balance of acid and bases in order to maintain the physiology of the body. So, for example, the buffer system in our body is called the bicarbonate buffer. So the bicarbonate buffer in the body basically is HCO3 minus, which combines with H plus ions in the body to maintain the correct pH. So we call this the bicarbonate buffer system. So H plus and HCO3 minus. So if you have too much HCO3 minus, the solution will become more basic. And if you have too many H plus ions, the solution will be more acidic. So the relative acidity or basicity of the solution is indicated by something called as a pH. Now, solution's pH is the negative base of 10 logarithm of hydrogen ion concentration of the solution. So for example, if a solution has a pH of 4, the H plus concentration is 10 times greater than that of pH 5 solution. You don't need to know this. You just need to understand what the number means. A pH of 7 is considered neutral, and a pH of less than 7 is considered acidic. So in an acidic solution, the number of hydrogen ions is greater than the number of hydroxide ions. And if the pH is greater than 7, the solution is considered basic. So in a basic solution, there are more hydroxide ions compared to H plus ions. A basic solution is also called an alkaline solution. Now, a pH scale in general is a logarithmic scale, and the pH scale represents a 10-fold change in hydrogen ion concentration, which you don't really need to know to calculate, but just understand that an acidic substance has really high concentration of H plus ions. And buffers, on the other hand, are chemical solutions that resist any change in pH. Now, in our body, we have a sodium bicarbonate buffer system, which help in maintaining the pH of blood, which is about 7.4. And this is crucial for physiological functions. Any fluctuation in this pH can be devastating to the internal organs. So it's very important that we have a good balance of acids and bases in the blood. So a lot of food we eat is acidic. So we have buffers to maintain pH, like the sodium bicarbonate buffer that we talked about. So water itself is neutral in nature, but most body fluids are near neutral, which is important for physiology. because the pH of the blood is 7.4. But a lot of things we eat is acidic. Like milk is acidic, banana, tomatoes, black coffee, vinegar, lemon juice, all of these are really, really acidic. But it's okay because the stomach acid itself are as low as pH 1 to pH 3. So it can fluctuate between pH 1 to 3. So the acid in the stomach can basically dissolve a piece of metal at times. But don't try that at home. Now, the reason our stomach doesn't get corroded is because our stomach is lined with a bunch of tissues that prevent our stomach tissues from being eaten up by the hydrochloric acid in the stomach. Now, a lot of things that we eat are also basic in nature, like egg white, bleach that we use for washing clothes, ammonia, oven cleaner, etc. All of these are basic in nature. Sodium hydroxide. Now, when you think of bases, they feel slimy, like the egg white is slimy, ammonia is slimy, sodium hydroxide is slimy, but that doesn't mean they're not corrosive. They can actually burn your hand enough to have a hole in your hand. So bases can also be really, really caustic and toxic in nature. So molecules can be divided into organic and inorganic molecules. Organic molecules are made up of mainly carbon and hydrogen. And inorganic molecules, on the other hand, include salts, metals, substances made from single elements, and any other compounds that don't contain a carbon bonded to a hydrogen. But in organic compounds, carbon is always attached to hydrogen and other substances. So depending on the type of attachments to the carbon, the chemical properties of carbon can be modified. So there are 4 categories of carbon compound. You have carbohydrates, lipids, proteins, and nucleic acids. Now, carbon has 4 valence electrons, meaning it has 4 electrons in the outermost orbital. So it tends to look for other chemicals to attain stability. So this affinity of carbon to form covalent bonding gives rise to more complex, larger molecules, and we call these molecules as macromolecules. So here is an example of methane. One carbon atom can attach to 4 other hydrogen atoms to obtain octet, and this in turn gives rise to CH4, which is methane gas. Now, keep in mind, organic molecules will always have carbon attached to hydrogen, but the backbone of carbon and hydrogen can attach to several other functional groups to give the organic molecule its chemical property. So functional groups are basically, so biomolecules are present in every living thing, and there are 3 main types of biomolecules: carbohydrates, proteins, and fats. And the 4th type of nucleic acid, which makes up the genetic material, which is also a biomolecule. Now we have monomers and polymers. Monomers are small molecules which add up to form larger polymers, which we call macromolecules. So macromolecules are very large organic molecules with really high molecular weight. And polymers are macromolecules made up of repetitive series of identical or similar subunits. So basically polymers are made up of monomers. Starch, for example, is a polymer of glucose, and it's made up of more than 3,000 glucose monomers attached together. Now, polymerization is the process wherein monomers combine to form a larger macromolecule we call as a polymer. So normally, when monomers combine to form larger molecules, it involves removal of water. So this process is known as dehydration synthesis. We call it dehydration synthesis. Dehydration means removal of water. Now during dehydration reaction, a water molecule is removed. So an OH of one monomer and the H of the next monomer is removed as a water molecule. Now, when macromolecules are broken down into smaller molecules, it involves addition of water. So we call this hydrolysis reaction. So during dehydration reaction, water is removed. So this involves removal of water from OH of one monomer and H of the next monomer. They combine to be removed as a water molecule. So this process is known as dehydration reaction. Then the resultant is known as a polymer. Now when the polymer is broken down, it involves addition of water. So we call this a hydrolysis reaction. So during hydrolysis reaction, water is added, and when water is added, the monomer is formed from breaking down of a larger polymer. So the first major macromolecule is carbohydrate. Carbohydrate is used for quick fuel and short-term storage of energy. For example, when you feel really tired, you want to drink something sugary, something sweet. You're going to have a banana, or you're going to have a sweet drink like Gatorade. You're not going to have something fatty like, say, a block of butter, right? So when people are really low on energy, sugar provides instant energy. So carbohydrates, which is nothing but sugar, is used for instant energy. Now, some examples of carbohydrate on a day-to-day basis include sugars, any starchy food like potato, banana. All of these are composed or are called carbohydrates. So carbohydrates have a general formula. It is called CnH2nOn. And every carbohydrate consists of carbon, hydrogen, and oxygen in the ratio 1:2:1. For example, the chemical formula of glucose is C6H12O6. Missing a 6 over there. So you can see that the glucose also follows the generic formula CnH2nOn. Mono means one or single, and saccharide means sugar. So monosaccharides are monomers that make up several different types of sugars, and the simplest form is called monosaccharide. The three most important monosaccharides are glucose, galactose, and fructose. Now these three are important, but you can see that they have the same molecular formula. So they're really the same sugar with different chemical properties. For example, glucose. You can see this difference between glucose and galactose. So this particular functional group, you have H on top and OH on the bottom. In galactose, this H and OH group is flipped. You have OH on the top and H on the bottom. So this subtle change in the placement of H and OH group makes them two different sugars. Fructose, on the other hand, also has 6 carbon atoms, where you can see that it has a CH2OH group on either end, making it a totally different sugar. But they all have the same molecular formula. Now, no matter what carbohydrate you look at starch, you look at plants, you look at the complex sugars in banana, they break down in the stomach into monosaccharides. Now, the simplest form of sugar is what is used to obtain energy. Now, disaccharides contain two sugar molecules. Di means two, saccharide means carbohydrate. So disaccharides are made up of two monosaccharides held together. So the three most important disaccharides include sucrose, which is nothing but table sugar. Sucrose is made up of glucose and fructose. So here you see glucose and fructose. Lactose, which is present in milk, is made up of glucose and galactose. Now maltose is made up of two molecules of glucose held together. Maltose is the white sugary dust you see on frosted flakes. So this is very commonly used in cereals. So oligosaccharides are polymers of monosaccharides. These include short chains of 3 or more monosaccharides. So at least 10 monosaccharides held together can be called an oligosaccharide. Now, if there are more than 50 monosaccharides held together in a chain, we call it a polysaccharide. So the most common polysaccharides found in nature are glycogen. Now, glycogen is a polymer of glucose, and all the excess glucose in the body is converted to glycogen in the presence of insulin. So insulin is a hormone that lowers blood sugar, but finally the sugar is converted to glycogen and stored in the liver. So when blood glucose is low, glucagon stimulates the liver to break glycogen back to glucose and release it into the bloodstream. And then starch is a polysaccharide we come across on a daily basis. Several different types of food contain starch. Now cellulose is a molecule, a polysaccharide that is found in plants, and human beings do not have the enzyme to digest cellulose. So cellulose in plants is just an important dietary fiber, but we cannot really digest the cellulose completely. Now this picture shows the structure of glycogen. Glycogen is stored in the liver, and any excess glucose in the body is converted to glycogen by insulin. So you can see that they are really long chains of glucose, and they are highly branched. So if you break down glycogen completely into monomers, the end product will be glucose molecules. Now the next important biomolecule is lipid. Now lipids are hydrophobic molecules with a very high ratio of hydrogen to oxygen. Now lipids are used for long-term storage of energy. Very common examples of lipids include fats, oil, and waxes, and these are hydrophobic in nature, so they do not dissolve in water. So these are hydrophobic in nature. Hydrophobia means water, fear of water, so they do not dissolve in water. And the lipids are used for long-term storage of energy. So in humans, we have 5 primary types of lipids. First one is fatty acids. Then you have triglycerides, phospholipids, and eicosanoids, which are a type of hormones, you can call it, and then you have steroids, which is used for synthesis of a lot of other things in the body, like cholesterol and several different hormones like estrogen and testosterone. So lipids are used for long-term storage of energy, and so everything that goes unused in the body is finally converted to fats and stored for the long run. Now, fatty acids are basically monomers of lipids, and you have two types of fatty acids: saturated fatty acids and unsaturated fatty acids. Now, saturated fatty acids do not have much variation between them, and many can be packed together, so they are packed with energy. So right over here you see a saturated fatty acid. Now saturated fatty acids don't really have too many bends or turns. They're just a straight chain of carbon and hydrogen with a functional group, which is a carboxyl group in the end. Now saturated fatty acids are solids at room temperature because you can see that they neatly stack over each other. And unsaturated fatty acids can be easily broken. So this picture over here shows saturated fatty acids. So you can see that these carbon-hydrogen chain, they just beautifully stack over each other. So at room temperature, they solidify. Now unsaturated fatty acid end up having a double bond somewhere in between, which causes them to have a kink or a bend. So right over here you see a saturated fatty acid with no double bonds, but the unsaturated fatty acids have a double bond between carbon atoms somewhere in between in the carbon-hydrogen chain. So this results in a bending of the structure of the fatty acid. And because of this kink, the fatty acids cannot stack together. So they are liquids at room temperature. Now, when you have a double bond between two carbons in between, they basically resist breakdown, so they are more unhealthy. So with respect to fats, you also have trans fatty acids and cis fatty acids. So trans fatty acids, you see a kink somewhere over here, and trans fatty acids are unhealthy because they are really resistant to enzymatic breakdown in the body. So we don't really naturally have the enzymes to break down trans fatty acids. Now, cis fatty acids contain two covalent single bonds in the same direction adjacent to the C to C double bond. So you have a CH bond over here, CH bond over here, and a C-C double bond right over here. So the cis fatty acids are found in nature very commonly. Now lipids are the next type of fats. Now a steroid is basically a lipid with 17 carbon atoms in 4 rings. And cholesterol is like the master steroid from which other steroids in the body are synthesized. Now steroids are important in this property of cell membranes. We'll look at it in chapter 3 and 4, but cholesterol specifically helps in maintaining fluidity of a cell and prevents them from breaking when the temperature gets too cold. Now, several different hormones like cortisol, estrogen, testosterone, bile acids, all of these are derived from lipids, specifically from sterols. Now you have good cholesterol and bad cholesterol. Now you have high density lipoprotein, which is considered good cholesterol because it has a lower ratio of lipid to protein. Now, low density lipoprotein is considered bad because it has really high concentration of fat compared to protein. So LDL is a major contributor to cardiovascular disease. Now, proteins are the next group of biomolecules. Now, protein is basically polymer of amino acids. So amino acid is the monomer of proteins. And several amino acids combine to form a polymer known as protein. So in general, the protein molecule is made up of several different amino acids. We have 20 amino acids in the body that are used to make larger proteins. Now, each amino acid contains an amine group, which is an NH2 group, and it contains a carboxyl group, and along with a radical group or R group, which is variable. But every amino acid will always contain an NH, a carboxyl group, and a C attached to an H atom. You can see this is a generic formula for an amino acid. You see an amine group over here, a carboxyl group over here, and a side chain R, which you can call a variable group for simplicity. Okay. So the properties of proteins are determined by the R group, which is a variable group, because this is the only group that is different in all 20 amino acids. For example, when R is replaced by CH3 or a methyl group, it becomes alanine. And when R is replaced by a phenyl group, we call it phenylaniline. So the R group gives each amino acid its chemical property and uniqueness. Now these are more examples of amino acids. Right over here you see glycine. Now glycine has a COOH group, so that's a COOH group. And then you have an NH2 group or amine group. And then you have a CH group, which is constant. The only thing that's different is the H group in place of the R variable group that we see. In the next group, phenylaniline, again it has an NH2 group. It has a carboxyl group, which we call the COOH group. It has a CH group, and this is the variable R group. Again, in aspartic acid, you have an NH2 group, you have the carboxyl group, you have the CH group, and this over here is the variable R group. Similarly, for phenylaniline, you have NH2, COOH, CH, which is constant in all of these amino acids. The only thing that's different is the R group. So this over here is the phenyl group. Now, the bonds between amino acid is called a peptide bond. So the peptide bond is unique to protein. And when protein denatures or breaks down, it breaks down into amino acid that we'll look at in just a second. Now, peptides are named by the number of amino acids. So if there are only 2 amino acids, you call it a dipeptide. If there are 3 amino acids bonded together, you call it a tripeptide. If there are about 10 to 15 amino acids bonded together, you call it an oligopeptide. And polypeptides have more than 15 amino acids held together, whereas proteins have more than 50 amino acids held together. So the bonds between two amino acids, so right over here, this is one amino acid and this is another amino acid. This bond in between the two amino acid is called a peptide bond. So the protein structure is encoded by the genetic material, and the simplest form of the protein structure is known as the primary structure of the protein. So the primary structure is the order of the amino acid as you see on the genetic material. And the secondary structure of the protein is a coil-shaped folded structure held together either in the form of alpha helices or beta sheets. So alpha helices are helix-shaped structure, and then you have beta sheets. And the next level of protein structure is known as the tertiary structure. The tertiary structure is formed due to interactions between the amino acids in the chain of amino acids. So the tertiary structure form the tertiary shape due to hydrophobic and hydrophilic interactions between the R groups of the amino acid. Now the last level of protein structure is the quaternary structure. The quaternary structure contains two different types of protein held together to form a bigger functional protein. For example, this picture is that of hemoglobin and myoglobin. So hemoglobin is basically a combination of two proteins, hemoglobin and myoglobin, which together form a big functional unit that helps transport carbon dioxide, oxygen, and nutrients in blood. So protein itself, keep in mind, is always the functional product or the final product of gene expression. Everything that's written on the genetic code is finally converted into the protein form to be used functionally. For example, enzymes in the biochemical reaction, or the keratin on your hair, collagen on your skin, is all proteins. We also use proteins in communications as hormones. We use it in bodily function like in enzymes. They help in catalyzing chemical reaction. We use it in the immune system for fighting infections and so on. So finally, whatever functional product in the body, in most cases, it is protein. All right. So the last part of the chapter is enzyme and metabolism. Now, enzymes are biological catalysts. They are biological catalysts, meaning the presence of the enzymes helps in increasing the rate of reaction. Without enzymes, biochemical processes would take place in an extremely slow manner, so cells wouldn't be as efficient as they are. Now the substrate is the molecule upon which the enzyme acts, and the product is basically the end product of the biochemical reaction. Now each enzyme is specific to a product. One enzyme cannot react with just any random substance in the body. So the enzyme is specific to its substrate. For example, amylase enzyme in the mouth or saliva helps in digesting starch only, and lipase is the enzyme that helps in digesting fats. So each enzyme is specific to its substrate, and they act by lowering the activation energy. So activation energy is basically the minimum amount of energy you require to initiate a chemical reaction. So this graph shows the lowering of activation energy in presence of the enzyme. So this is an example of a reactant, something that needs to be converted to another form. Okay, so the reactant has a really high energy level, but in the presence of enzyme, the activation energy gets lowered and it makes the process or the reaction more efficient. So enzymes are biological catalysts which enhance or increase the rate of reaction. Now, enzymes can only function at certain optimum conditions. For example, enzymes in the stomach can function only at an acidic pH, whereas enzymes in the intestine or enzymes in the blood function more at a neutral pH of 7.4. Enzymes also get denatured in certain conditions like extreme acidity or basicity or extreme temperature. We call the process as denaturation when they can no longer function as an enzyme. So they basically get degraded. So this picture shows the action of an enzyme. So an enzyme is specific to a substrate. So this is an example of sugar hydrolysis. So the substrate is sucrose or sugar. The sugar attaches to a region on the enzyme known as an active site. And once the sugar attaches to the active site, it forms something called an enzyme substrate complex. And the enzyme substrate complex leads to a chemical reaction that breaks the bond between the sucrose molecules, and in the end, glucose and fructose are obtained as the end products of the reaction. So in this case, sucrose is the substrate, sucrase is the enzyme, and glucose and fructose are the products. Now sometimes enzymes need some additional help, and that help is provided by cofactors and coenzymes. Cofactors are inorganic molecules that assist the enzyme, for example, minerals like magnesium, calcium, iron, and copper. And coenzymes, on the other hand, are organic molecules like vitamins that assist enzymes in biochemical processes. So the next biomolecule is nucleic acid. So there are several types of nucleic acids in the body. The main types are DNA, which is deoxyribonucleic acid, RNA, which is ribonucleic acid, and then you have ATP or adenosine triphosphate. ATP is the energy currency of the cell. So the main function of nucleic acid is to store, transport, and translate genetic information. So DNA is the nucleic acid that makes all the basic units of hereditary material, which control the cellular structure, cell activities, and even gene transference, or the characteristics that we inherit. RNA, on the other hand, is a different nucleic acid. Structurally, it's different because it only contains a single strand normally. And in the process of protein synthesis, DNA is first converted to an RNA form, and RNA is made into a protein. So the flow of information from DNA to RNA to protein is known as the central dogma of the cell. So the monomer of DNA is known as a nucleotide. So nucleotides are basically biomolecules that form nucleic acids. And a nucleic acid is a chain of repeating monomers. And each monomer consists of a 5-carbon sugar, and it contains a phosphate group, which is an inorganic molecule. Then it contains a base, which is a nitrocarbon ring. So the main difference in DNA and RNA is that DNA is deoxyribonucleic acid, whereas RNA is ribonucleic acid. DNA contains 2 deoxyribose sugar in the second position, and RNA consists of just a single ribose. Another main difference in the DNA and RNA molecule is the nitrogenous bases that make up DNA. Now, DNA is made up of thymine, adenine, guanine, and cytosine. Now, in an RNA molecule, instead of thymine, it contains uracil.